Now that we have introduced the general mechanisms and processes that drive early development, let's consider how evolution relates to developmental biology and how the field of developmental biology has contributed to our understanding of evolutionary relationships among species. Something that has been observed is that the embryos of different species have similar structures, but those embryonic structures don't always give rise to the same tissues and organs in the adult organism. For example, the pharyngeal arches are structures that are present in the embryos of vertebrates, but in the fish, they give rise to the gill arches, in reptiles, they give rise to parts of the jaw, and in humans, they give rise to bones of the middle ear. However, in general, a germ layer typically gives rise to the same type of tissues and organs in different species. So, for example, skin and the nervous system are derived from the ectoderm, and this is something that applies across species. The relationships among species can be established by finding common embryonic and larval forms. Similarities in embryonic structure suggest common descent, and organisms are actually classified based on their embryonic and larval forms. For example, barnacles and shrimp are both classified as arthropods based on the features of their larval forms, even though as adults these organisms look very different. This suggests that there are generalized features that appear early in development, while more specialized features appear later. This was recognized by Darwin, and the relationships between evolution and developmental biology have been described as four interconnected principles known as Bombeur's laws. The first principle states that there are general features that are present in a large group of organisms, and these features appear earlier in development as opposed to the features that are characteristics of a smaller group of organisms. For example, the notochord, which is shown here, is a structure that's of mesodermal origin, and it's present in all vertebrates. I should also note that the notochord is also present in chordates that are invertebrates, like the tunicate. Now, this particular structure arises very early in development in all chordates, which all have the structure. More specialized structures like forelimb bones adapted for flight, which are present in some but not all types of vertebrates, appear later in development. Because general features that are characteristic of a larger group of organisms appear early in development, that means that the organisms belonging to that group, in this case vertebrates, for example, will appear very similar right after gastrulation, and that's what's shown in these drawings. The second generalization, which is related to the first, is that early in development, structures derived from a particular germ layer look similar among species. However, the specializations of the individual groups of organisms within a larger class appear later. For example, early in development, vertebrates have the same type of skin. As development progresses, structures like scales, feathers, and hair begin to emerge in the organisms belonging to different groups. So the embryo of a higher species does not pass through the stages of development of the embryos of lower species. Even though the pharyngeal arches look very similar in vertebrates early in development, these similarities fade over time. So the pharyngeal arches in humans never pass through a fish-like state. It follows from this that the embryo of a higher animal will never look like the adult form of a lower animal. In other words, a human embryo is never going to look like an adult lizard. Instead, and coming back to the first point, the embryos of different vertebrate phyla appear similar to each other right after gastrulation, and this stage is known as the phylotypic stage. At this stage, there are similarities among vertebrate embryos, both at the anatomical level but also at the molecular and cellular levels. Phylogenies are often assembled using morphological characteristics, in other words, looking at similar morphological traits. So that means that it's important for the morphological similarities that are being used to establish or to determine evolutionary relationships to actually be based on common ancestry. In other words, those characteristics used to classify organisms should be homologous. Homologous traits are traits that are similar because of common ancestry. An example of that is the structure of vertebrate forelimbs, which have similar bones. On the other hand, if traits have a similar function but arose independently of each other as a result of convergent evolution, they are said to be analogous. So these traits are similar because of their common function, not relationships in ancestry. For example, the wings of birds and the wings of bats are analogous as wings because they arose at different points in evolutionary history rather than from the same common ancestor. Bats arose from a non-winged mammal. Now, although a bat and a bird wings are analogous as wings, they are actually homologous as forelimbs because the structure of the vertebrate forelimb arose from a common tetrapod ancestor. To provide another example, the wing of a butterfly and the wing of a bird are analogous. The two share a common function, which is why they're both called wings. However, they did not arise from a common ancestral structure that became modified through evolution into bird wings and butterfly wings. Homologous anatomical structures can diverge as a result of changes in development, and these alterations in development provide the variation that's needed for evolutionary change. Bats and mice are both mammals, and their forelimbs look similar early in embryonic development. Webbing is present between the digits, and it's important for separating the digits. But in mice, this webbing is lost once it has served its function. In bats, there are alterations in gene expression that prevent the death of the cells responsible for the webbing. Additionally, there are other differences in gene expression that cause the digits of the forelimbs of bats to grow longer compared to those of mice. And this illustrates how evolutionary changes in anatomy take place through changes in development as a result of alterations to gene expression.